Next up, we will have Dr. Phil Empey from the University of Pittsburgh, who will be presenting on the All of Us experience. Phil.
Thanks, everyone. I really appreciate the opportunity to share some of our work with the All of Us program. I'm not sure I'm super excited about being in the regulatory section, but I think hopefully there'll be an additional lessons learned for this. I think it'll be a little bit different from the last two, perhaps, because just from the very outset, this is a research study, and therefore the paths are different. But I think there's some interesting observations and some lessons learned. Many of us, of course, are doing research as well as clinical, or a blend between the two. This is the path that this study takes, and it may be some mirroring things in some of your projects as we proceed. Alrighty, I hope everyone is aware of All of Us, but All of Us is a large national NIH-funded program that endeavors to create a cohort of at least 1 million diverse participants across the U.S. With the goal of advancing precision medicine. So where we were at about a year ago, and this is advanced from here, of course, is data on that number you see here, so many hundred thousands of patients. Data has been released on about 245,000 whole genomes, short-read sequencing, as well as array data, as well as EHR data and participant surveys, as well as some demographic information available for research. It's also unique in that it endeavors and has accomplished returning genetic information. So at the very outset of the program, this was a very high-profile goal for the reasons you see here. One, at the very outset, it was very clear that it's important health information, right? It's also at the same time, along with many other sort of organizations pushing that participants who participate in research deserve to have their results returned. There was a significant National Academies report, as well as increasing pushes in academic spaces that this wasn't our data to withhold from participants, but something that should be shared and could be engaging, and it was deserving of the folks who are participants in accomplishing research. In the lay world, at the same time, anyone can go into a CVS pharmacy or on the internet and essentially do some degree of genetic testing, whether it's health related or not, but certainly a lot of folks are very aware of this and actually seeking this outside of provider or health systems. The other part of this was the expectation that the more we engage participants, the more we return value, the more there'll be participants, and so that was a core philosophy of the program. So return of results equals return of value, although it is a research study to keep in mind. So why pharmacogenomics? I sort of don't have to preach to this group, but the important thing is the study actually asked very early in planning numerous focus groups to say, if we were to return genetic results, what would you be interested in? And pharmacogenomic test results were sort of equivalently at the top with disease risk genes, with a lot of response from the lay public saying this would be important to us. Tell us whether our medications are going to work, and this data seems like it's important. So it became a priority from the very beginning, from 2018, even before funding of the genome centers, that we would want to do this. Some unique caveats about the design of the program, though. You can kind of follow the steps down there. Like a traditional research program, consenting, of course, enrollment sampling, biobanking, some organization doing testing, and then, in this case, a clinical validation laboratory to be able to translate those findings, and then results returned to a participant. What you'll notice is there is no provider in that last group at all. No pharmacist, no clinician whatsoever. It was a direct return, and therefore more akin to something you might see with a DTC test, and certainly raises some flags of how we would do this safely and appropriately within a research study. The model looks something like this. I don't know if any of you participate in the study, but after signing informed consent, you actually have the option to provide secondary consent to be able to essentially access your genetic data. So you can say no, of course. You can say yes. This is the first stage in that profile, and then once you pass it and go through this step, you'll be able to select what types of genetic data you may want to receive. So in this case, pharmacogenomics and hereditary disease risk are separate decisions, and each informed independently and then agreed to independently before any type of report is generated. Again, that is triggered after the participant elects for that. So in this model, though, it was decided pretty early on, and this was an interesting decision, that if there was a positive disease risk association because of the dominant model that was in clinical practice at the time, that these results need to be notified to the participant and then returned directly via a genetic counselor. Non-informative results could be accessed directly. And in this point, clinical confirmation was offered. So this was performed by a clinical lab, and then the results provided back still to the patient directly. Pharmacogenomics results were treated differently. They were treated perhaps because of the scale or the accessibility or dominant practices that many of us are doing in our current roles, that the PGX results, once signed out and requested by the participant through consent, are released directly and available for download directly. The counseling resource is certainly available and pushed to say if you want to talk to someone you can, but it was not required. And we might imagine a number of reasons why, not only sort of what was going on in clinical practice at the time, but also because the positivity rate, if you want to call it that, would be nearly 100% with any certain number of genes because of the high frequencies of actionable variants. So we will talk about regulatory. For this particular group, of course, it is a clinical trial, and the study's sponsor is NIH. So in the investigational drug exemption process, that role is important because that role gets to decide what path people take and how things are triggered through the IRB and through the FDA. Obviously, those groups have critical roles as well, but the criteria and the design is determined by the study sponsor. IRB plays a key role. You can see what they are responsible for. They do have to review everything that is created, of course, to assess risk, and they bring in a diverse group to be able to assess that. The FDA's role, you can see listed here. We've already talked about it. It was decided very, very early on to engage with the FDA out of the gate from the study sponsor directly to the FDA because of the visibility, the size of the program, and the scale. Frankly, it hadn't been done before, and we wanted to get all individual viewpoints. The other thing that was important to understand is the testing that is being done achieves CLIA standards. It is done in CLIA labs. All traceability aspects of things is to that standard, and the data generation is in genome centers that meet that CLIA standard. In terms of interpretation and secondary confirmation, again, same rules apply, but it is important to distinguish this from a regular clinical test, so there was no supervising clinician that ordered the test. There was no clinical test requisition, and the sampling handling logistics for this study are quite complex. You can imagine all across the country, everywhere from clinics to institutions to within pharmacies to within trailer recruitment centers that may go out into rural populations. Samples are all transitioned and sent to the biobank, which is at Mayo, and then routed out into the individual 3 genome centers at Broad, Baylor, and University of Washington. So this is a scary slide. This is the timeline. It took about 19 months from the first engagement with the FDA to the IDE being approved. You can see there's a lot of spaces in there where a lot of things were being discussed and trying to figure out. It started off with the very first understanding that the FDA believed what we were doing is a significant risk device study and therefore must go through the IDE process. And then there was a series of a lot of pre-sub queries where everything was discussed. The design of the program, how things were done, where they're being done, how results were returned, ideas for return, what was going to be on the reports. Essentially, all these went through a lot of individual discussions with the FDA. The green lines are the verbal interactions. Some of these were through formal meetings. The blue lines are written actions back and forth, and then, I'm sorry, the gold is the other written action. You can also see that once you submit something, there's a set time for response, and you have a lot of quick back and forth at the end as you approach approval. So I'm just going to call attention to some of the PGX specific ones because we binned the activities in trying to understand analytical validation and then the return aspects into a variety of different pre-subs, and then getting to the point that we understood those things and be able to advance. Now, initial discussions with us were with the device group, so CDRH within the FDA, but then I'm very pleased to report, and it was super helpful, that midway through the process in the PGX discussion, CDER was brought in. and provided excellent support, I guess, to understanding the landscape and providing direction on some things. So that was very helpful and I think led to our ultimate success. So again, 19 months altogether, and we did achieve that approval in mid-2020. So some big lessons here, just discussed in the last presentation. For this one, they made a clear distinction that there was no clear predicate device. 23andMe was brought up. Some of the EnSite programs for a number of years ago were brought up. But this was different. This was viewed differently, and because of where testing being done in multiple different places and how the return happened, there was not a clear comparator. They also made the distinction that the device is not just the part that was happening in the lab. It was in the genetics lab, it was the entire thing, end to end, from sample collection all the way to the report, and all parts of that had to go through review. The other things that may be different, or not totally different, but kind of a lesson learned from this, is the FDA strongly preferred patient-derived samples over any type of reference or cell lines as ground truth. So I was doing comparisons, and you can say, well, I can order some cell lines from Corel or other places. That was not the preference. In most situations, we had to try to find clinical samples from the existing clinical labs we already had on hand that were orthogonally validated or we already had Sanger information on. The other thing that's interesting that I find kind of curious versus other types of genetic testing or other maybe applications of genetic testing, specifically in inherited disease space or cancer pre-screening, the FDA did ask us to demonstrate performance at every individual reportable gene and allele. So many of you who may be aware of testing, what often happens in sequencing is you create a bedded region, it includes the entire gene, and then when you come back, you say your allele, your coverage depth across the entire sequencing exceeds 98%, so that part of the confirmation is acceptable. In this situation, although it's still from sequencing, we were asked to demonstrate success not across an entire gene, but at every individual variant that we thought was important to be able to call an individual allele, which I would argue is a significantly higher bar than what is done in CLIA-based environments or even large sequencing-based testing. The other thing is just to keep in mind, I'm going to say it like many, many times as part of this, and they did as well, we were going through the IDE process. So the end result of this is a research investigational device where results can be returned to participants, but we did not solicit and request any type of assertion regarding validity or utility, and therefore it is not labeled as such. So at the end of the day, the report says investigational device only, and whether you believe the analytical validation is exceptional or not, it cannot be used in clinical care without clinical confirmation by that regulation. So I'm going to walk through what our end result was, and this is in 2020. Some things have changed since, and some things are in the middle of being changed. So when we decided what genes we wanted to report out of the gates, this was very early in the program, and the pharmacogenomics world has changed a bit in the last 6 years with new guidelines, new genes, new associations coming out. But initially we chose these 7 genes because we were thinking about the clinical return specifically. We were avoiding use cases where there is significant routine clinical monitoring, where there may already be more challenges in the risk association. You can think of things like warfarin, tacrolimus, maybe, where you may already have some monitoring going on in clinical practice. We also were thinking about the breadth of perhaps getting the maximum exposure, opportunity to provide value to our participants. There is a gene on here that is missing that everyone always says, where is 2D6? That had to be delayed somewhat because of the more complicated requirements for analytical validation, right? We had to have structural calling in the pipelines. And with this program, we were very much building it as we went. And at the beginning, there was no structural calling in the pipelines at all. So how we decided to pick criteria, and this is a little bit different than perhaps how AMP would do it in the tiering guidelines or how you might do it your own lab, or but maybe not. I mean, just you have to think about the patient population that was being recruited and understanding the goal was diversity and to understand and sort of characterize some of that diversity. So we did focus on alleles with known functional consequence. We didn't want to spend our time and energy and efforts trying to validate allele that had normal function or uncertain function. We did bring in standards from Tier 1 and Tier 2, so both of those with AMP, but we went a little bit beyond because we did not use the minor allele frequency cutoffs. We were hoping to find things that were exceptionally rare as long as they had function, and tried to put the effort into validating those genes if there were controls available. So we did then go to FarmVar and pull the necessary variants. That was a critical resource. Again, there is a focus on looking for rare alleles, and then it's specific to diverse populations. And then the big one is we had to find things that had controls. So without controls, we can't do the validation. Without the validation, we can't return. So there is a big wish list, and then there is what we can do now. And the goal was to be able to expand that as more controls, either from clinical sequencing or even found within the program and then brought back for orthogonal confirmation, could then lead to enhanced recruiting or validation. So this is the results of the validation, and then this is all published. So I feel free to sort of pull and go through this extensive paper that explains both hereditary disease risk and the pharmacogenomic process for the IDE. But you can see the performance was excellent across multiple genome centers, between labs, within labs, accuracy, sensitivity, precision, all exceeded the metrics necessary. We were focusing on blood-derived samples because that's the majority of the collection, but it also do work with saliva samples. The scope of this was large. I mean, it was over 1,200 samples, had to be processed at multiple facilities and multiple times to be able to achieve the standard that was necessary for the submission. So this was the initial variant allele list that met validation standards that were able to be confirmed. We didn't have any that we went for and couldn't get. These are the ones that we had controls, and these are the ones that were included, again, 2018. So please think of that as you're looking at this. Some things have changed. For example, 1B1, the SAR17 allele, no longer exists, right? So I already saw that in the front row, yes. And I was on that too, so yes, we did have to make a pivot after the IDE, so we can talk about that later too. But you know, the world does change, and a 19-month process is not very amenable to rapid changes. Everybody is very aware of this. We did use the full CPIC translation tables and did submit those along with the validation process in order to get approval. So just contrary to prior things in the research-based process, that was accepted. From the reporting of drug-based associations, the program wanted to provide information on drugs. But there's no provider involved, and we learned very quickly that it was not going to be perceived as acceptable to put any CPIC recommendations on a report, even if that report said, please bring this to your provider, because the chance of misuse of the data without an understanding of the data was deemed exceptionally high. But the perception that you can't claim medication associations in an IDE process is also not true. We have approval, and if those of you got your results back that way, if you're in the study, know that there are drugs on this report, but there are not recommendations along with those drugs. So we believe that providing drug information is a good trigger, and that was what was submitted. So it was predicted phenotype to drug, and if the predicted phenotype was found in the individual participant, the drug was listed. We did consider medication factors such as route of administration, and when we had to weed through some of those. And again, this was very iterative back and forth, specific questions on specific genes with, hey, this is what we're proposing to submit, is this acceptable? And then, you know, responses back and forth in the formal process. Again, big shout out and benefit for CDER and sort of bringing in the expertise needed for pharmacogenomics, I think, during this evaluation. Not expecting anybody to read this, but this is sort of our internal rubric of how we approach whether we wanted to list a gene-drug association before it got approved or not. And I would love to get to the point that this statement was just accepted and we didn't have to challenge every single one of them, but we are not to that point yet. But we did bring in the FDA product labeling directly if it was in those categories. If it was in section A or B, the first two sections of table associations, pharmacogenomic associations from the FDA, as well as CPIC specifically. And we took the higher of all of those. So oftentimes when CPIC said something but it was not in the other two, we advanced the CPIC recommendation. So let me pause here a little bit. This was informative. This was at the time that we just heard about, and it was a lot of, they'll never approve that, they'll never approve that, they'll never approve that. We got approval for almost everything we submitted. There were a few instances where data was only pharmacokinetic. I can think of maybe some of the TCAs as an example, where the CPIC guidelines made some sort of broad statements around thinking because of the change in this, Our author group believes it's clinically relevant, and the FDA disagreed and wanted clear clinical outcome data, which does not exist yet. So in those, some of those were not approved, but the majority of them, the escitalopram, the sertraline, those were all approved. PPIs approved, they're on the reports. So there has been a lot of things that may not be as front and center in labeling, original labeling, that's on that table or in a CPIC guideline that was acceptable through this process. So just to sort of get through this, what it looks like at the endpoint, this is a screenshot of a report. This is just the page one. We did go through full user comprehension testing, similar to what 23andMe did, for making sure that it was understanding with diverse patient populations, multiple round of focus group testing. This was done by the CVL partner led by Color, is the one that did this, is funded to do this for the study. You can see what the goals are. The overall goal was to trigger conversations with healthcare providers and route people to have those conversations. So please discuss with your doctor and pharmacist. Pharmacist is all over that report, which was a win. It does say very clearly, this is a research result. You can see it sort of right across the header. It's on every single page. It does encourage sharing, includes normal results, and it emphasizes that genetic information is just one piece, as well as risk mitigation statements like, if you're prescribed this medication, keep taking it and talk to your provider about these results. So it did get through these rounds successfully. We did have to change a bunch. There were things that you might not have expected, like medicine versus medication. When someone hears healthcare provider, they think insurance company, not a physician. So we had to change some words. Pharmacogenomics, you can imagine, is not stated on that. And if it is, it's just kind of like in quotes and said that's what this means, but the title of the report is Medicine in Your DNA, in order to make sure there was some good understanding. So we learned a lot around the language that needs to be in these reports to achieve that user comprehension bar. So just because I want to mention the genes, so and drug association. So this is the simple listing of what it looks like. It lists brand and generic along with the gene if you have the particular phenotype where the association was approved. And that's it. So it deliberately says if the medication's on the list, do not stop taking it. Have a conversation with your healthcare professional. So where are we at now? We initiated returns in fall of 2022, and we're well beyond 225,000 have been notified to date. So in terms of results being ready for access and return. We are working on expansions and plans for updates. I'm not able to share any of those really just yet, but you can probably imagine what genes we want to add at this point, including expanding sort of the coverage and updating things as well. I did put 2D6 on here. It is not a promise from the organization. I can't speak for the organization, but it is something that many of us have been pushing if it's capable from short read sequencing, and we will find out as we go through the validation for that. And we expect things will change. There is some discussions around whether we need to have enhanced data available for clinicians to bring to the providers, a resource, or whether we can direct them to the resources we're all aware of. So I'm sure this will be revised as research comes around this return of results model. So takeaways from this, I hope you get from it, is that obviously research-based return is heavily regulated by IRBs and the FDA and the program sponsor, whoever that research organization is. Sometimes those discussions for IDE are made at the IRB level. I have some studies that were made that way, but sometimes it's made by the study sponsor at a higher level. Either way, you have to adhere to the FDA standards if you're going to go through that process. You may have seen these results in your practices. If you see them in your practice, the party line is you may not use them for clinical care. So our institution, our policy says don't scan them in because they're not a clinical result. But when we get them, we have great conversations and we direct folks to repeat clinical testing, and I have yet to see one that hasn't confirmed. You can imagine from the bar of the analytical validation, that may be unexpected, but again, must be clinically confirmed. Just a final plug, and maybe this isn't the best meeting for it, but I do want to emphasize that this community has also really been looking forward to and hoping that the All of Us program would get to having more haplotype calling available. So the researchers in the group, these data are now finally available. A couple weeks ago, we released the data from a demonstration project, and all the haplotype calls for Stargazer and FarmCat are now available to the research community. We'll have some training coming up at the PGR meeting in September, and our hope is to make these data available on the resources you all know and love as well, and we're working towards that. So that is all I had.